Could be morning.
Join.
You guys are. Anything less than 15 minutes, 1 minute.
Down the blonde page. These young boys don't know what the dawn made. I'm just a bad boy gone clean. I'm the guy changing. Always remain so. Never drinking. All you know this man made me that come from. Being free to love, rolling back to back in line.
All right, good morning. Can you hear me? If you can hear me, please type 1 in the chat box.
I hear you.
All right. This Zoom can only accommodate 300 people. So I'm just going to send the YouTube link to the WhatsApp group so that the class reps can also send it to your groups. All right. I think we have 300 people already. So I've just sent the YouTube link. Can someone please confirm that those watching via YouTube can hear me? I just want to do a bit of test running so that we will all be on the same page. Okay, I can see that some people are watching.
Nothing for you, bro. Nothing for you. Not.
Please, if you're not speaking, you're not asking questions, just stay muted. Or maybe I should just mute all of you, except that once in a while. So maybe we'll just use the chat box because it's a large class and I might not be able to control all of the distractions. Right. Okay. 35 people are watching on YouTube. Right. So I'm just going to share my screen in a moment. Can you confirm that you can see my screen? Please use the chat box. If you can see my screen, type 2 in the chat box. 2. Good. Good. Okay. Please, I need somebody to also help me check those on YouTube. Can you confirm that they can see my slides as well? Those on YouTube. Please don't write your name in the chat box. We don't need attendance. Zoom is going to give us attendance, and I think I might be able to pull out some names as well from YouTube. So it's fine. It's a large class. We don't need attendance today. Okay, so those on YouTube can also see my screen. They can see my slides. Right. So we're just going to start. Maybe I should start recording. What time is it? I think we have like 2 minutes more. All right. I'm just going to click the record button now that we have tested everything and we are sure that everything is working. Great. Okay. Good morning. Can you hear me? I think you can all hear me. Yes. All right. I've started recording. So my name is Dr. Vodering Otu. I'm a lecturer at the Department of Cell Biology and Genetics. And it's my first time, teaching BIO101 since I joined the university. I think Dr. Adebeshi taught this part last year, and thankfully, I had access to his slides, and I used some of his lecture content. And I'm saying this because in the academia, plagiarism is a big crime. So some of the things on my slides are also from his lectures, right, from last year. I think that some people's videos are on. I'm supposed to stop video. Where is that setting now? I should have done it during the. Okay, let me just hide video panel. Yep, I think so that it will just not distract me. Okay, so I have said that I used some of the slides, some of the content from Dr. Adebeshi, who has been teaching, this topic for several years, and you would find some of these contents, in my own lecture slides. At the end of the lecture, I'm going to send the slides to your course rep, so you only need to jot the most important things. I'm going to be saying a lot of things that you might not find on my slide. So I expect that you have a note and a pen beside you, that you would use for this lecture. Please, one of the class reps, once it is 8:50, should let me know so that I would begin to wrap up, because I could talk, for 2 hours, and I understand that you have another lecture at 9. Right, so we're just going to dive in. So we're looking at cell structure and function. My email address is also here. If you have any questions that we're not able to answer during the lecture, you could write me an email. I would try as much as I can to answer within 3 days. That should be like the max. So at the end of this lecture, we expect you to understand the various cell types, the cell theory, the concept of the cell theory. We want you to be able to describe the structure and the organization of cells, as well as you must be able to explain the functions of cellular organelles. We have various cellular organelles. What are their functions? What do they do? You must be able to explain, the function of each of these organelles. And then lastly, maybe during my last lecture, we're just going to dive in briefly into photosynthesis and cellular respiration. We're just going to scratch the surface of this topic or this aspect, because you're going to be doing botany, where you will be taught extensively. Even this year in year 1, I think BIO108 or something like that, or 102, maybe in first semester or second semester. So they're going to go in depth, into this topic, but we're just going to scratch it on the surface, because this is, of course, biology. And some of you are only going to do biology just this semester. You might not have the opportunity, later on. So from secondary school, you already know that the cell, if I ask any of you, what is the cell? Everybody, or I expect that everybody should be able to say, oh, the cell is the basic structural and functional unit of a living organism. And that's correct. That is, the truth. I remember that I attended one interview and somebody said that I could use an example of a brick, right? So if you think of a brick, bricks that are used to build walls, in that brick, you have cement, you have sand, you have water, all of these things are mixed together, and then you have a brick. So there are so many things, basic units of that brick that makes up the block that you can see, right? And so that's the way it is. So your body, and we're going to look at the levels of organization. So you have cells that form the basic structural unit, the basic functional unit of all living organisms. And of course, this term cell was first coined by Robert Hooke in a publication called Micrographia in 1665. Sure, nobody on this call, was born at that time. And so he referred to this term cell when he viewed cork tissue, through a crude microscope. And this figure, maybe I should also use my pointer so that you can. This is just the cover of the publication, by Robert Hooke in 1665 called Micrographia. And, the word cell is actually from the Latin cellulae, meaning small room, small compartments, because at that time, these small compartments look like monk cells. These guys were monks, and, from the monastery, it was able to, describe it based on the description of the small rooms, that monks occupied at that time. And so he called them cell. And like I said, he viewed cork tissue, which was from an oak tree called, the scientific name is Quercus suber. And, the outer back was what he examined, and he was able to, get some pictures from the thin slice of that material. Some of you who might not have seen like the cork, if you think of your wine bottles, that cork on it, it was just something like that. A thin slice of it, but the back which is also called phloem, not phloem. Now, phloem is what you're familiar with, which is like the inner part, but phloem is what he observed at that time. And so, after that observation, of course, there was the concept of the cell theory, the unified cell theory. and 3 of them, proposed by different scientists. Matthias Schleiden, who was a botanist, worked on plants, and in 1838, together with a zoologist known as Theodor Schwann, who was, of course, working with animals in 1839, proposed the first concept of cell theory. And they both said that all living organisms are composed of one or more cells. So you could have unicellular or multicellular. And that was their concept of the cell theory, which again, I believe that you're familiar with. Again, they also said the cell is the basic unit of structure and function in living things, which is perhaps one of the most widely accepted definition, of the cell, and it's part of the unified cell theory. But the third one is that all cells arrive from pre-existing cells. And this, of course, has to do with the concept of cell division, right? So Rudolf Virchow, or Rudolf von Virchow, in 1855 stated that omnicellular acellular, which means that all cells arrive from cells. So if you think about it, you evolved from, or you're a product of your parents, right? And it's the same thing. So every cell has a parent cell, and then in biology, we say things like daughter cells, right? So all cells come from, previously existing cells. And that corrected Schleiden and Schwann's earlier ideas about spontaneous cell formation. Because I just wanted you to look at the pictures of some of these guys, which I got, of course, from the internet. But these were the people that defined the cell theory, which, 3 of them have been widely accepted as postulated by them. So we look at the levels of organization in biology. Again, I'm sure you're familiar, from atoms to biosphere. So you have molecules, macromolecules, cell, tissue, organ systems, or organ system. And then you have organisms, population, community, ecosystem, biosphere. And in this course, I think that you're going to look at some of these levels respectively. So I am focusing on cell, but I know that later on, I think Dr. Dada or Professor Onjoko might be coming in to teach you ecology. And most of the things they would look at, would be the concept of population, communities, ecosystems. These are things that you look at. But if you look at the level of organization, you see that there is an increasing complexity from atom. Atoms basically are the smallest unit of an element. So you have things like carbon, lithium in this figure or picture. And then you have molecules. When 2 or more atoms combine together, you also have macromolecules. So we talk about proteins, which are things that we're also going to look at, in a moment. Carbohydrates, lipids, even DNA, they are macromolecules. They are very large. They are complex molecules that are essential, for life. And they also have small subunits, small units, which we call monomers. And then you then look at the level of cells, which we have defined as the basic structure, structural and functional unit of a living organism. So you have unicellular organisms, you have multicellular organism, but at that level, we look at cells. But even before cells, some authors mention organelles, because organelles, but again, cell is all encompassing, because within the cell, you have various cellular organelles. But organelles are just basically structures within a cell that have specific functions, and we are going to look at several of them in this topic. But we know that tissues, they are collections, a group of similar cells that work together to perform a specific function. So you could have nervous tissues, you could have, various tissues, muscle tissues, and these are collections of cells, and then for organ, the same thing, collection of tissues, working together for a common function. And then you have systems, multiple organs. So for example, the digestive system, respiratory system, having different organs that perform, some functions. Then you have an organism, which is a complete individual living being, right? It's, a complete living being, and that's an organism. Then you have population, you have communities, you have ecosystem by itself. But these are just the levels of organism. But for this topic, I am particularly interested, and I'm going to delve into cell. And that brings us to the definition of cell biology. What is cell biology? In cell biology, we study various cellular processes carried out by individual cells, such as cell division. I don't know if you have been taught, but I think that in this course as well, you'll be taught mitosis and meiosis. So in cell biology, we're looking at cell division. Remember, we said that all cells come from previously existing cells. So how do these cells divide? How do they grow? How do they reproduce? We're also interested in organelle inheritance. How are organelles inherited? So for example, and maybe we're not going to delve too much at this level, but those of you who take cell biology in year 2, you'll be taught the endosymbiotic theory, which explains how mitochondria evolved, how the chloroplast also evolved. So again, in cell biology, we are interested in organelle inheritance. Please note that some of the things I'm saying, questions might come from there. So you might not see these things on your slide, but please note them. And of course, this lecture is being recorded, so I could also go back to the lecture, the recording, watch it, and then take a few questions. So these concepts, of organelle inheritance, biogenesis, how are organelles generated, synthesized ab initio from the beginning. So again, those of you that are studying cell biology, or I think some of you in microbiology usually take some cell biology courses as electives, you're going to look at membrane biogenesis, how are membranes synthesized, for example. So these are things that we are interested in cell biology. Signal transduction, how are signals sent? How do cells communicate? And then cell motility, how do they move? I'm sure that many of you are familiar with various locomotory organelles. If you've done Zoo 101, and even in secondary school, high school biology, you've heard of flagellum, you've heard of cilia, which we're also going to look at. But these are some of the things that we're interested in cell biology. How do organelles move? Organisms move, or cells, how do they even move? How do they migrate? Which locomotory organelles do they have? So there is what we call the actin-dependent motility. We also have like the flagella-dependent one, which of course has to do with the activity of the flagellum. So these are some of the things that we are interested in cell biology. So how do we visualize cells? You know that cells are very small. You cannot see them with the naked eye. So cell biologists require and rely on microscopes. So in your practical, I think one of your labs, you're going to have access to a light microscope. Maybe you're going to handle them, I'm not sure, because your class is quite large. But you're going to see what a microscope is. I think maybe you are going to have like access to a compound light microscope in the lab. But we use these microscopes to visualize cells because you cannot see cells with your naked eyes. When we're working in the lab, you have to be very, very careful. You just see, sometimes depending on the color of your medium or something, but you don't know the state and the well-being of these cells until you view them under the microscope. And we have different types of microscopes, light microscopes, of course, various examples, a fluorescence microscope, dissecting microscope. There are different types of light microscopes. So you have fluorescence, you have dissecting, you also have compound light microscope, which is also a type of light microscope. But of course, from the name, you know that there is a light source. You're going to also see the components, various components. And if you also check your lab manual, I think that lab manual, even before I prepared my notes, I also took my own copy of your lab manual, my lab manual for this course. And, I reckon that there are lots of valuable information that you would find in that manual. So you would see the various parts of a microscope labeled, their functions. That's not for me to delve into because it has already been extensively explained in your lab manual. So please do well to read it up. But you also have electron microscopes that can resolve structures that are about 0.2 nanometers apart. So, sometimes some of the images, which we call micrographs, are generated from electron microscopes because they also have like higher resolution. So for compound light microscopes, you have two or more lenses, which we call the objective lens and the eyepiece, or ocular, right? So you also have transmission electron microscope, but basically we just use different types of microscopes depending on what you're working on per time. And I'm going to show you the next slide. Of course, this is just a figure of a compound light microscope, yeah? But I used a fluorescent microscope. So I actually looked for the original picture of the microscope I used. I really couldn't find it, but then I just inserted this one. It's fine because the idea is just to show that you require a microscope to view cells. And these are cells from mice skin. They're called stem cells. Later on again, you're going to be taught pluripotency, totipotency, all of these kind of things, how stem cells are formed. But just have it at the back of your mind that these are stem cells that I worked with a few years ago. And we were able to add some factors from my skin, and these cells were produced, into what we call embryoid bodies, EBs. Embryoid. Embryoid is spelled E-M, like embryo, EMBRYOID, embryoid bodies. And these cells here were viewed on day 2, of the experiment. But by day 5, so again, this slide is doing 2 things. I'm trying to talk about how you visualize cells, how you see cells. Let me stop this one. But by day 5, so because it's a video, by day 5, you can see that these cells have grown. This is the same cell from day 2 to day 5. You can see increase in cell morphology. So cell size, basically, that's what morphology is. And cell size is limited, actually. As cell size increases, it takes longer for materials to diffuse from the cell membrane to the interior part of the cell. So again, in cell biology, we talk about the surface area to volume ratio, right? And so you can see that by day 5, these same cells on day 2 have now grown bigger. But by day 7, so again, this slide is going to do the third thing, which is to explain the concept of differentiation. Because by day 7, these stem cells have differentiated into cardiomyocytes, which are cells of the heart, and you can see them beating already. So, like I said, these are cells from mice. They are mouse-induced pluripotent stem cells, and they have now differentiated into cardiomyocytes that are beaten by day 7. So just like the way your heart cells beat. So, but we couldn't have seen any of this without a microscope. And these cells they are expressing green fluorescent protein. That's why they are green. It's a fluorescent dye, that helps to visualize. So it's also a marker such that it is only cardiomyocytes, that you would see expressing these green fluorescent dyes. So, of course, there is a bit of genetic manipulation, designing your construct and all of those things, which at this level, you shouldn't be concerned about. But what you just need to know is that you can observe cellular changes, cell morphology. Sometimes we even want to take pictures of these cells and then measure the size using softwares like ImageJ, to measure like diameter. So you monitor growth. So sometimes you introduce a compound, you want to see if it will affect the cell size, if it will affect the morphology of the cell, right? So you want to measure in plants biology as well. Sometimes even till now, there is what they call morphological characterization. They use your ruler to measure like the leaf size, all of those things, the diameter. Again, I mean, of course, you can argue that that's not at the cellular level, but even at the cellular level, we are interested in the cell size, the diameter, and you can use some softwares, to monitor all of this. So, but you cannot see, if I showed you the plate, and maybe, I mean, I could have just even showed like the plate from which these cells were observed. You cannot see any of these things from the plate. All you just see is a pink solution or maybe an orange solution, which is the color of the medium, that these cells were given. Of course, cells also have to be given nutrients, just the same way you eat, right? So these cells also have to be given like growth medium for them to grow. So all you just see is just whitish patches or white patches, on the plate, and that's it. But by the time you view under the microscope, you are able to see these cells beating or not beating, for example, depending on what activity you're trying to monitor. So there are many, many things that you can look out for. And then you can also see cell deaths. So sometimes you see some cells that have died. And you know this because, for example, we have what we call the Ginster stain. So many procedures in cell biology, but by the time you view under the microscope, you can also see things like contamination. So sometimes you see bacteria, bacterial contamination, and then at that point you have to discard. There is a procedure for discarding cells, cell culture, in the lab and all of those things. So, but you cannot see any of this except you view under the microscope. So we move on. There are two types of cells, basically, based on the presence or absence of a nucleus. You have eukaryotic cells and prokaryotic cells. And from the name, you know that eukaryotes have true nucleus. They have a well-defined nucleus. They're found in humans, animals, in plants. But prokaryotes, you can say false nucleus. And it's actually coined from the Greek word pro, meaning before, and karyo, meaning nucleus. So when you hear things like karyorexis in second year biology, again, I'm preparing some of you. I know some of you might not come back, but even those of you going to medical school, you hear things like that, karyorexis, karyolysis, all of those kind of things. When you hear karyo, it has to do with the nucleus, right? It has to do with the nucleus. And so eukaryotic means true nucleus, while prokaryotic means they do not have a distinct nucleus. Their nuclear material is just lying freely in the cytoplasm, in what we call the nucleoid region. That's what it is called. So they do not, prokaryotes, apart from not even having a distinct nucleus, they also do not have membrane-bound organelles, such as mitochondria and the plasmic reticulum. And examples are bacteria, cyanobacteria, which are blue-green algae. They also have like archaea as well. But eukaryotes, the examples are specified here. I can use my pointer again. Okay, so it now brings us to the domains of life. So from I'm just laying like foundation because it's important that we have this foundation. So do all stem cells? Okay, I'm just going to take questions at the end because I can even see that I've spent almost 30 minutes and I only have like 3 lectures to give. So I'll take the questions maybe once it is 8:45. Somebody should please let me know by 8:45 so that we'll take questions, because if I start answering questions, it might slow us down. So we have 3 domains of life: archaea, bacteria, and eukaryota. So archaea and bacteria would represent prokaryotes, while eukaryotes, of course, they have, like we said, a distinct nucleus, well-defined nucleus, and outer membrane-bound organelle. So if you look at this figure, you would see the various examples of bacteria. You have cyanobacteria, you have tamotoga, you have proteobacteria, gram-positives, green filamentous bacteria. And then if you look at the archaea domain, you see various examples: Metanococcus, you have allophiles, which of course they are salt lovers. and then if you look at eukaryotes, you see fungi, plants, which are things that you will look at, in botany, in zoology. For example, you have different flagellates, trichomonads, and all of those other animals broadly. But these are the domains of life. And like I said, bacteria and archaea do not have a distinct nucleus. That's why they are called prokaryotes. They also do not have many membrane-bound organelles. And I said many because, for example, in bacteria, you would find ribosomes, right, in them. But ribosomes do not also have membranes. So generally, prokaryotes lack membrane-bound organelles, while eukaryotes have membrane-bound organelles. And so this is the figure of a typical bacterial cell. And like I said, you can see the ribosome here in the cytoplasm. This is the cytoplasm. You have the ribosomes, which do not have membranes. They also have, sometimes they have plasmids, which are circular DNA, right? They have plasma membrane. They have membranes, right? Which we call plasma membrane or cell membrane. They have cell wall. Bacterial cell wall is made up of peptidoglycan, right? Unlike plant cell wall, which is made up of cellulose, or fungi cell wall, which is made up of chitin, right? Bacterial cell wall has peptidoglycan, and that's the composition of bacterial cell wall. And so you see the ribosomes in bacteria, in prokaryotes. I mentioned cell motility the other time. So you see the flagellum here. This is the nucleoid region, which has the circular DNA, which is, the genetic material in this, in prokaryotes. So the ribosomes in bacteria, looking at some similarities and differences compared to the ribosomes in eukaryotes, is smaller, right? So it has two subunits. The first subunit is 30S. S is a unit, is like a unit, like an SI unit. So when you have seconds, the unit of time. So S means Svedberg, and I'm probably going to type it in the chat box. Svedberg unit. So 30S and 50S. So if you add both of them together, you're supposed to get 80. But in prokaryotes, we say they have 70S together instead of 80. Whereas eukaryotes has 40 and 60, and then we say they have 80S instead of 100. So please note this. They have Svedberg, and Svedberg unit has to do with their sedimentation coefficient, right? Their density of the ribosomes when they are centrifugated. So you put in a blender, you blend, the sedimentation coefficient of the small subunit is 30S, while that of the large subunit is 50S for prokaryote ribosomes, for prokaryotic bacterial ribosomes. But for eukaryotes, you have 40S for the small subunit and then 60S for the large subunit. So like I said, prokaryotes generally, they have a nucleoid region. Their genetic material is lying freely in the cytoplasm. All right, so this is just a figure of eukaryotes, animal cell and plant cell, which many of you have drawn severally in secondary school, and again, I took these figures from Dr. Adebeshin's slides, like I mentioned at the beginning of my lecture. He also had like other figures, of animal cells that showed the cilia, and other locomotory organelles. But if you look at this, you can see various organelles. You look at the nucleus, and the structure of the nucleus, the nuclear pore, the chromatin, nucleolus, nuclear envelope. You look at centrioles, you look at intermediate filaments, which has to do with the cytoskeleton, is part of the cytoskeleton. You have microtubules, microfilaments and intermediate filaments. Three of them make up the cytoskeleton. Please don't forget microtubules, microfilaments, and intermediate filaments. They make up the cytoskeleton. And so here you can see intermediate filaments. You also have the plasma membrane. You have the Golgi complex. You have small vesicles, in the cell as well. You have the mitochondrion, ribosomes, free ribosomes. When they are also attached to the endoplasmic reticulum, you have the rough endoplasmic reticulum, and then you have lysosomes. I said earlier on that you have microtubules, then you have smooth endoplasmic reticulum. These ones do not have ribosomes attached to them. We're going to look at the functions of all of these organelles individually. So, but in plant cells, you also have some of these organelles that I mentioned. For example, the nucleus is there. The mitochondrion is also there. But in addition to the mitochondrion, they also have chloroplast, which is a type of plastid. There are other types of plastids, which we are going to look at, in the course of the lecture. They also have like cell wall, which is a major difference. So a question could be that you should differentiate between plant cells and animal cells, which I expect you to already know, because these are questions that you have been used to and been answering even from high school. So they have cell wall, and like I said earlier on, their cell wall is made up of cellulose. So you might want to compare and contrast between a plant cell and an animal cell. Maybe that could be like an assignment for you guys. I'm not going to mark it, but just have like a table of maybe 5 to 10 differences between and similarities. Maybe we should do 5 similarities between plant and animal cells, and then 5 differences in terms of organelle composition. Now, all right, so let's just move on. So this figure kind of even helps to answer the question I just I just asked you to do as a form of assignment. But it's the cover page of our year 2 manual in cell biology, although it's also the figure of a plant and animal cell. But this figure, why I like it and why I included it, is that it shows the various organelles that are present in both animal cells and plant cells. And then it gives a brief description, a very short, memorable description of the function of some of these organelles. For example, ribosomes are protein synthesizing machines. Very simple, straightforward, easy to remember. Peroxisomes destroys peroxides. Cytoskeleton supports the cell. So in terms of cellular structure, aids in movement of organelles, although there are many other functions, excuse me, of the cytoskeleton, right? Because sometimes they serve as tracks for vesicles, to move, for molecular cargos to move, within the cell. But like this explanation, they support the cell, they help to give like some form of structure, and then they help for the movement of various organelles such as vesicles, So you look at so many of them, which when you get the slides, of course, we are still going to touch some of them one by one, but for some of you, I just felt that it might be easy to remember. It's like, what do we call these small past questions that we used to use then? Short answer question. No, it's not short answer. Key point or something like that. I can't remember the name now. Yeah, key point or something like that. Yes. So we had things like that. So this is more like the key point, for this for various organelles. So if you look at plasma membrane, it says it separates the cell from the environment, regulates movement of materials into and out of the cell. So, if you want to have a general and holistic view of various organelles and their functions, briefly summarized, this figure is something that you should consider. All right, let's just move on. Key points, yes, thank you. So I found this recently and I thought to include it because it kind of gives you an idea of the volume, occupied by major intracellular compartments. For example, in a liver cell, so this is a hepatocyte, which is the cell of the liver, right? So if you look at a liver cell, you see that 54% of a liver cell is made up of cytosol. 22%, you would find mitochondria. So sometimes in cell biology, when we want to isolate mitochondria, we work with liver homogenates. So you get mice, and I'm trying to give you like a mental picture of these things. So you have mice or rats, we have experimental animals. Some people also use rabbits, and then you remove, when you sacrifice the mice. We don't say we kill mice in science, we say we sacrifice them. So when you sacrifice these mice, you excise or remove the liver, and then from that liver you can isolate mitochondria. There are protocols, which at this level you shouldn't bother your head about because you're not going to do it in the lab. But maybe in year 2 or year 3, some of you that will continue will now begin to isolate the mitochondria, from mice liver. But again, from this, you see that 22% of a liver cell, has mitochondria. Then you have the rough endoplasmic reticulum, 9%, nucleus about 6%. You have other organelles like peroxisomes, lysosomes, endosomes, all of these percentage, and then if you add them up, they would make up about 100%. So this is like the composition, although it differs from cell to cell depending on the cell need, right, of the cell. So let's look at the cytoplasm, because if you look at this, I mentioned the cytosol, which is which is the part of the cell that gives like the largest volume, for example, in these liver cells. So, but the cytoplasm consists basically of the cytosol and various organelles. So when you talk about cytoplasm, which is the first part of the cell that we want to look at, it's the internal volume of the cell, exclusive of the nucleus. So when you talk about the cytoplasm, it is the cell minus nucleus. Cell plus nucleus is what we call protoplasm. I'm sure you also know this. But for those who may have forgotten, protoplasm is the living content of a cell. It just comprises the cytoplasm and the nucleus. So, but when you say cytoplasm, you are talking about the internal volume of the cell without the nucleus. So various organelles plus the cytosol, which is jelly-like, like a semi-fluid in which organelles are suspended. And so this cytosol is found between the cell membrane and the nucleus. They mainly compose, or they are composed mainly of water, salts, and various organic and inorganic compounds. So both bacterial cells, plant cells, and animal cells have a cytoplasm. So if you look at the figure I showed you earlier on, you can see the cytoplasm here. If you look at this animal cell, you can also see the cytoplasm. And then if you look at the plant cell, where is the cytoplasm here? Anyway, all of these components is not labeled here, would be the cytoplasm where the organelles are. That would be the cytoplasm. I'm trying to see if this figure also has it. Okay, so but I just wanted to point your attention to the fact that both bacterial cells, plant cells, and animal cells have. So a typical exam question could be, which of the following is true? Or one of the following do not have a cytoplasm. A, bacterial cells, B, plant cells, C, animal cells, D, none of the above. What would the answer be? Type it in the chat box. What do you think the answer would be? A, bacterial cell, B, plant cells, C, animal cells, D, none of the above. Just use the letter A, B, C, or D. Or yes, none of the above, D. Great. So, I mean, those are the ways to look out, for questions and to, to answer them. Right. So you have all cells possess a plasma membrane, cytoplasm, genetic material, and ribosomes. Again, this could be like another question. So both prokaryotes and eukaryotes, they have plasma membrane. Remember I said, and if you look at the figure of the prokaryote that I showed earlier, you would see the parts labeled plasma membrane. All cells have a membrane, or we also call it cell membrane. So sometimes, or some textbooks call this plasma membrane cell membrane. Sorry, I'm not able to look at the charts in YouTube because I am using my laptop, and if I minimize my screen going to YouTube, I don't have two screens. Now I know why some people have two screens, in the bank or even in some universities abroad, but we'll look into it because I could have opened YouTube on the left and then to see their comments, but we'll check it later on for those on YouTube to see your answers. But I'm assuming that you're also saying none of the above. Anyway, all cells have a plasma membrane. They have cytoplasm. They also have genetic material, although, like I said, it could be lying freely in the cytoplasm. They may not have a membrane-bound nucleus, like prokaryotes, which have just a nucleoid region, or plasmid DNA, which is like circular DNA lying in the cytoplasm. And then there are ribosomes. I also told you, they have ribosomes. Prokaryotes also have ribosomes, but in terms of the size, the sedimentation coefficient, the ribosomes in prokaryotes are 70S. The ribosomes in eukaryotes are bigger, they are larger. So you have 40 and 60. The other one you have 30 and 50. Right. So let's move on. So we'll look at the plasma membrane. And the plasma membrane is an essential feature of every cell. It defines the boundaries of the cell and its various internal compartments, ensuring that its contents are retained. So every time I teach this topic, because in year 2, there is a topic called models of the membrane. It's this topic, or this part of this lecture is actually a full topic in year 2. So those of you doing BIO101, by the time you get to second year, those in maybe biology education and other departments that will be taking that course, you'd see that this topic is a full topic on its own. But whenever I teach this topic in year 2, so we can have up to 40 slides on this slide. So, but again, in this lecture, I think there are only 3 or 4 slides, just because we wanted to just summarize and give you like an introduction. So this is introductory biology. But when I teach this topic in second year, I try to describe the plasma membrane as the gates of the University of Lagos. So those of you who have passed the first gate or the second gate, which is at DLI, you would observe that without going through the gate, you cannot say you are in the University of Lagos. You can be outside, around, maybe at Emerald Hostel or Abule Oja, until you enter the gate. If somebody called you, that's the only time you'll be able to say, oh, I'm inside Unilag, right? Okay, it's 8:45. All right, so I'm just going to stop in the next 2 minutes. Let me just wrap up my thoughts. So that's when you're going to say that you are inside Unilag. So the gate serves as a boundary, right? If there is a problem outside the university and the university security department decides to lock the gate, every one of us remains inside Unilag and we cannot go out. That's what the plasma membrane is. It basically serves as a boundary. What is wrong with my slide? As a boundary. So if you look at this black line around here, that's what most people refer to as the plasma membrane. But this black line around here is actually not a line, as we will soon see. It is made up of lipids, proteins, carbohydrates. Because if you look at the structure of the plasma membrane, you talk about a phospholipid bilayer, you talk about so many things, and it has various functions. But one of the function, like I said, is the boundary, boundary function. So it serves as a boundary that controls what goes into the cell and what goes out of the cell. So if you think of a gate, you can be right. If you call the plasma membrane a type of gate, that controls what goes in and what goes out. So we're going to look at the other functions, in our next class, but I want to try to answer some of the questions that was asked. Please, can you put the questions again now in the chat box, so that I won't have to be scrolling too much? If you can copy some of those questions for me and paste now. And for those on YouTube, I don't know if somebody has access to 2 devices and there are 1 or 2 questions that you think will benefit the class, you can copy for me. Please also put them now in the chat box. Is plasma membrane the same as cell wall? No, plasma membrane is not the same as cell wall. So cell wall, you have cell wall in prokaryotes, you also have cell wall in plants, right? They give structure, and shape. But plasma membrane, you can find in both plants, in bacteria, in prokaryotes, in eukaryotes, and they also have like their own functions. 5 of them, which we'll look at in detail in the next class. But the answer is basically no, they're different. Plasma membrane is the same as cell membrane. So if you call plasma membrane cell membrane or generally membrane, you will be fine. In fact, apart from having a cell membrane outside the cell, every organelle or most organelles, except for 1 or 2 of them, which we will look at, examples of some organelles that do not have membrane, but most organelles also have their own membrane, that helps to localize the function of that organelle. So all of the things, depending on the structure of the organelle. So some organelles even have 2 membranes. So organelles have membranes, but the cell also has its own membrane, which we call the plasma membrane. Let's look at the second question. This is there are many messages now. Is bacteria a prokaryote or a eukaryote? Bacteria is prokaryotic. So what is the sedimentation coefficient used for? It's an SI unit. It's an SI unit. So usually when we want to isolate various organelles, right, it has to do with the density, of the organelle. So you put some of this liver tissue, for example, if you're working with the liver, in a blender or what we call a homogenizer, and then you blend it or homogenize it, and then you have what we call tissue homogenates. It is in that tissue homogenates you have your cells, and by the time you add a couple of reagents and you do centrifugation, you spin, there is a protocol. I don't know the protocol for isolating ribosomes, but at the end, based on the density and the sedimentation, that is, it will settle down. Based on that, there is a sedimentation coefficient. For small subunits, it will be 30S. The SI unit is Svedberg, and that's what that sedimentation. So the small subunit will sediment at that 30 Svedberg unit, while the larger subunit, it has 2 subunits, 2 components, like A and B, together to form 1 ribosome. So the smaller part is 30S, the bigger one is 50S in prokaryotes. Let's see the next. These questions are many. Sir, please, can you explain the 70S? I think I just explained it. For eukaryotes, you have 40 and 60 as the small subunit and large subunit respectively. And then for prokaryotes, you have 30 and 50. Is bacteria? I've answered this one. What are the Svedberg units? I've answered it as well. Is the plasma membrane the same as cell wall? I've answered this. What is this? Why are the same questions come? What is a cytoskeleton? So it's a part of the cell that gives the cell structure. It is made up of, so you think about it as like your skeleton in the body. So cyto from cell. So anytime you hear cytology, you're talking about cells, cyto, right? Cytoplasm, cells, cytology, cells. So cytoskeleton is what gives, just like your own body, you have skeleton. It gives your body, the framework, the structure. So there are three types. You have microtubules, microfilaments, and intermediate filaments. Again, in year 2, it's a topic on its own, and then you're going to look at the structure of microtubules, the size, the functions, the drugs that can affect them, so many other things. But for now, they also help in terms of cell division. So you know that when you say chromosomes align on the equator, some of the things that help the chromosomes to align, you talk about centrioles, you have intermediate filament, microtubules. That is the cytoskeleton. Please, sir, what is 50, 40, 30? I think I've explained that. What are these bed bag? I've explained that. Is it the same as cell membrane? Yes. Bacteria is prokaryotic. Yes. Please, sir, I didn't get this concept of sedimentation. I think I've answered this. How many subunits do? I don't know if I'm reading the same questions though. Cytosol is the fluid part of the cell. They do not have. I think I've answered all these questions. The jelly-like, what is a bacteria? It's not, yes, it's not done via normal arithmetic. So one would assume that 40 plus 60 should give 100, but no, it is 80s. And then the other one, instead of saying 30 plus 50, it is 70s. So please note this, because that could also be a potential question. Sir, you mentioned that the microtubules, and one other one. Please, can you go right? That's the intermediate filament. So microtubules, microfilaments, and intermediate filament. Peroxisomes. I said they destroy peroxides, right? So you have sometimes, in the cell, you have free radicals. You have free radicals. So when you're talking about oxidative stress, right? So there is an organelle that helps to remove some of these free radicals. And again, in cell biology, we look at aging. So the videos I showed you just explain the concept of cell differentiation. So we also have what we call senescence, which has to do with aging, senescence. So cells will grow, they will old. It's like you. And that's why sometimes when you grow old, you begin to have gray hairs. So it's the same way with some cells. They grow old, they look old. We call them senescent cells. And then eventually they die. They can die through apoptosis, which is a programmed cell death. The cell is programmed, destined to die, or necrosis, which is usually due to some of the chemicals of physiological injury. What else? Is there any other question I've missed? Cell wall and yes, how was the answer none of the above, sir? I don't know the question here. Alix way. How does visualizing cells work and how many things can you see under the microscope? You can see a lot. Depending on whatever is going on in the cell. In fact, now we have what we call single cell transcriptomics. So people sometimes are not even interested in several cells. They are interested in working with only one cell. They're interested in working with only one cell, and with some microscopes, with AI, artificial intelligence, you can even cut out one cell. So sometimes you can work with a cancerous cell, and then you look at the cell population, you look at the signatures, immune signatures, peptide signatures in just one cell. So a microscope helps you to do all of that. So what's the lifespan of a cell? It depends. For example, red blood cells is 120 days. So it depends. Different cells have different lifespan. But when they grow old, they eventually die. Yeah. Why does an animal cell not have a cell wall? Because they do not need it. So sometimes, even some organelles, the more organelles you have in a cell depends on the cell need, the need. So if the cell needs it, it would have, these are things that people have studied based on the structure. Every scientist has studied, various animal cells, and they have not seen cell wall there. made up of cellulose. So if an animal cell does not need it. So please, are there any organisms smaller than a cell? I don't know what you mean by organism, because when I talked about the levels of organization, you cannot even have organism. Do you understand? So if you look at atoms to molecules to macromolecules to organelles to cells, after cells, you then have tissue. After tissue, you have organs. After organs, you have systems. So that's the level of organization. So I don't know what you mean by organism, but what you can have as a lower level of organization would be maybe macromolecules, atoms, and molecules, if that is your question. Please spell out the unit of that. It is Svedberg, Svedberg. It's Svedberg. When the cells are being renewed, why do people body look old? Okay, can you propose your cell theory? Well, yes, you can, but you have to have a lot of experiment to back it up. If you have something contrary, you can. But in science, we don't just propose. You must back it up with evidence, sufficient scientific evidence to say. In fact, I'm going to also mention some new organelles that have been discovered, recently by some scientists. Flexibility is important in animal cells, and they need cell-to-cell interaction. So cell wall. I'm trying to read your text. The messages are too. So cell wall could limit this. Yes. Thank you, Timmy. When the cells are being renewed, why do people body look old? Well, aging in itself is another topic on its own. In fact, one of my students presented on telomeres shortening. This is advanced biology. So aging is a topic in maybe year 3 or year 4 that we can look at. So you look at the chromosomes. At the chromosomes, you have what we call telomeres. So telomeres shorten. Why do telomeres shorten? What are the proteins? It's a big topic, right? So cells renew, yes, but aging also occurs. And there is what we call cell homeostasis. So there is sometimes an imbalance between the number of young cells compared to older cells. So as you grow older, aging occurs. There is telomere shortening. Your chromosomal ends shorten, and that leads to aging. That's a full topic in year 2 or year 3 that we can look at when you get to that level. So does this mean I can define apoptosis? Yes, apoptosis is programmed cell death. Please, could you drop the slides? I would send them at the end of the lecture. Maybe not today. So please I do not get what you said about the death of cell naturally and biochemically. So apoptosis is programmed cell death. I talked about cell growth. I said cells can grow, they can divide. That's cell division, mitosis and meiosis. I talked about cell differentiation. One cell can differentiate into different cells or even different organs. For example, your stem cell can differentiate into red blood cells, can differentiate into heart cells, can differentiate into various cell types. And then that's the concept of cell differentiation. Then you also have senescence, cellular senescence. When cells grow old, aging, and then you now have cell death, which is apoptosis or necrosis. These are the type of death. Apoptosis is programmed cell death. The cell is programmed, destined to die. There is a pathway, or there are pathways for apoptosis that we cannot cover also in this lecture. The time for this class is up. You guys have a lot of questions from YouTube. So how about the hepatocytes? Do they also die, even though the liver, all cells grow, divide, and die? All cells. Please note this. In fact, it can come as a question. All cells must grow, all cells must divide, and all cells die. So liver hepatocytes also die. Sometimes you have infections, when you have malaria parasites, some of these protozoans now, right? They infect the liver cells and then they destroy the cells. Sure, you understand? So sometimes you can have like invasion. You can have an external factor that would, affect the cells. Flexibility is important in animal cells. Okay, I think we said this. All right, time up because you guys have another lecture. So thank you so much. See you on Wednesday.